we go. Welcome, everybody. Thank you for your patience as we get everybody on board and everybody in the meeting room. It's great to have you back. Thank you so much for staying with us over these 4 days. We hope that you found this an inspiring course, an engaging course, and we've got a lovely feedback form for you to give us all your ideas and input at the end of today's session. So thank you so much on behalf of both Acilia and H3D Foundation. We really want to thank you for being a part of this workshop and joining in and giving us your energy and your time. These are such important tools and we really want them to work for you. So part of today's session, in a moment, I'm going to hand over to Miguel, and he's going to take you through the session today, which is going to answer a lot of your questions and also look forward into what the next steps are. We're going to end off today's session with a reflection exercise, which I will lead in terms of, which will give you the opportunity to set some goals for yourself going forward in terms of implementing this knowledge that you've gained. So we hope you enjoy this final session, and over to you, Miguel. Thank you so much.
Thanks, Nicola. Hi, everyone. Please let me know if this works. I'm sharing the screen now. Are you all seeing my screen?
Yeah.
Looking great.
All right. Good morning or good afternoon, everyone, and welcome to the last session. Today we'll do a little bit of an overview of what we have been seeing over the last 3 days. And perhaps most importantly, we'll also look into the future or what's next in AI for drug discovery. The focus is going to be in this field called generative chemistry, which you might have heard about, but we will also look a little bit beyond the materials of this course and basically offer a little bit of a perspective of how do we think Ercilia and H3D drug discovery with AI is going to look like in the next 5 to 10 years. So you have been a little bit exposed to this tool that we are currently building. It's in the making. It's called the Ercilia Model Hub. And the Ercilia Model Hub contains, as you have seen, different types of models that are able to do different types of predictions for your molecules. We have only seen just a tiny portion of it related to the prediction of antimicrobial activity of compounds, as well as other interesting features like toxicity and ADMET, but there is much more to that. So the first thing we are going to do today is to offer like kind of a broader overview of the tools that you can find in the Ercilia Model Hub if you are interested in using it. And of course, as always, if you guys have doubts, if you guys have questions, if you really want to use it in your future research, you should definitely reach out to us so that we can understand your needs and help you out. So the Ercilia Model Hub at the moment contains almost 200 models. That's a lot. We've been building this tool over the last 3 to 4 years, and we have organized these models in a kind of a taxonomy that hopefully is going to help us understand what kind of things you can do with the tool. The first type of tasks that we have in the Ercilia Model Hub, we can bucket them under this umbrella called annotation. When we talk about annotation, it's basically what we have been doing here in the course, is trying to predict properties of the molecules, right? These properties can be basically bioactivities. It can be physicochemical properties, like, for instance, solubility of the molecules, and it can also be bioactivities, right? For instance, growth inhibition, toxicity, etc. This is mostly what we have been seeing in the course, right? Given a molecule, trying to predict their properties, either physicochemical properties or biological properties in the assays. We have another type of tasks that we have also kind of touched, even if a little bit tangentially in this course, which is what we call molecular representation tasks. In the molecular representation tasks, basically what we try to do is to featherize compounds, that is to describe the compounds using fingerprints, embeddings, descriptors that was briefly introduced in the previous days, as well as project these representations into typically a 2D space so that we can visualize how those molecules are distributed in the chemical space. And I think that some keywords here would be like PCA or UMAP. Then finally, we have another type of models, which are the sampling models. When we try to do sampling, this means like trying to sample the chemical space. That is, given a molecule, an input molecule, trying to sample more molecules that might behave similarly or better than our input compound. When we do sampling, we basically have two types of subtasks. One is perhaps very well known to you, is similarity search. Given an input molecule, try to find similar compounds to my input molecule in other databases. And we also have generative models, right? So this is trying to predict new compounds using AI tools, compounds that are not in any databases, compounds that are genuinely new. All right, is this clear to all of us? Can you give me perhaps a sign if you are following? Is there any question in the chat? No? All right. All good. So let's start like going bucket by bucket and trying to offer at least one example so that you understand what we are talking about. Let's start with property calculation or prediction. There's many instances of that. For instance, solubility is something that people ask about all the time. But also one thing that people ask a lot is synthetic accessibility. So we do have models in the Ercilia Model Hub related to predicting or calculating the synthetic accessibility of compounds. This ID that you can see here is one of them. There's more. And the synthetic accessibility, I think it was discussed already yesterday, is a score. We have different types of models that have different types of scores, and each score is interpreted differently. The one that I'm showing here is called SIVA score, and it's a score that goes from -infinite, showing, as you can see, compounds that are really not synthetically accessible, to positive scores plus infinite, showing compounds that are really like plausible from a medicinal chemistry point of view, right? So this model in particular, you can use it to, given a SMILES string of a compound, try to assess whether it's going to be easy to synthesize it or not in the lab. There's more, and you should definitely browse the Ercilia Model Hub to see which other synthetic or retrosynthetic accessibility models you can find them there that you can find useful. All right. The next bucket here is activity prediction. We've seen a lot of those, right? We've seen activity prediction against Acinetobacter baumannii. We've seen activity prediction for cytotoxicity, for instance. I'm just showing another one here for antimalarial activity prediction. This has the model ID EOS4ZFY. And this is a model that it's interesting because it was built based on multiple data sets from pharmaceutical companies, right? So just want to show you here how are we actually evaluating whether those molecules, those models are interesting. Here we have taken a data set that we produced where we have like known actives and known inactive. And then we are basically calculating the score of this model in the known actives and known inactives in an antimalarial assay. And as you can see very clearly, is that the known actives tend to receive higher scores than the known inactives. Now, some of us might say, hey, but there's many that are actually not discriminative. That's expected. Models are not perfect. You have to remember that many times we build models on data sets that are not exactly, or they're not match exactly what we have in the lab. And therefore, some kind of error is expected in the model. So there is no clear separation, but what's clear here is that whenever we use these models, compounds that receive high scores, for instance, above 50, tend to be active more than inactive, right? So this model, if we apply a relatively high cutoff, it's actually quite precise. Yeah, might not have a high recall because we are still missing some known actives, but it's quite precise in that most of the molecules that are active tend to receive a high score. Okay, very good. Now, let's shift to representation. This is a little bit more abstract. This is a little bit more difficult to understand, but it's crucial if we want to do machine learning with chemistry. And the first type of models, the first family of models that we have in the Ercilia Model Hub are related to featurization. We do have many ways of featurizing compounds, and I think that this is something that sometimes is a little bit difficult to understand. So I want to pause a little bit and show you this image. I don't know if any of you have seen this picture previously. Probably not. This is a painting from the surrealist, and what's this painting saying here? I don't know if you can speak French. Many of you do, definitely. It's basically saying this is not a pipe, right? But it's interesting, right? Because it is a pipe. But what the author is saying is this is really not a pipe. This is a representation of a pipe. This is a painting of a pipe, right? This is a pipe. We are trying to depict a pipe in a language that we humans can understand, right? So our task here when we are featurizing compounds is to depict a molecule in a language that computers can understand, right? So this is not a molecule. This is a representation of a molecule, right? This is a representation that we humans can understand, but it's not a molecule. It's just a drawing of a molecule. The same way, we need to find ways of representing compounds so that the computers can handle them. And this is something that was already introduced previously and is related to this concept of molecular descriptors. And we have many types of molecular descriptors, and each of them is useful for different scenarios. It was commented previously, I think, in some of the questions that some people were interested in capturing the stereochemistry. In that case, you might want to use 3D descriptors, molecular representations that capture the 3D dimensions. Sometimes we want something more simple, and we just can use, for instance, 0D or 1D descriptors that capture more or less the 2D properties of the compound, right? Interestingly, in the last years, we have been developing descriptors that are a little bit more data-driven. These are molecular representations that have been learned from massive data sets using AI and machine learning tools, the same way ChatGPT has learned language by exploring the language that is available in the web, right? So these types of data-driven descriptors are extremely powerful, and we are incorporating many of them in the hub. I'm going to focus in particular in one type of descriptor that we have developed that is called the chemical checker. I'm going to expand a little bit upon it later on. Why is it so important to have good featurization? Well, because the machine learning task in drug discovery is always composed of two steps, as you have seen. The first one is converting the SMILES or the molecular structures into these numerical representations that the computer can understand. And the second step is converting these numerical representations into a prediction of bioactivity, right? So we have the featurization step and then the training step, right? The training step is actually what needs data. This is what where we need to provide the training data, right? When we are doing supervised machine learning. And therefore, the better our molecular representations are, the less training data we will need. All right, I hope that's clear. If we have very good molecular representations, very dense, very informative molecular representations, then probably we will need less training data to build our models. And because definitely drug discovery in the academic setting is data poor, it is very important that we invest a lot of effort in having the best possible numerical representations for our compounds so that we can train good models using as little data as possible. Is that clear to everyone? Yeah? Very good. So what is our approach to all of this? Our approach to all of this is to using these so-called chemical checker descriptors. The chemical checker descriptors, basically what they try to do is they try to capture first the chemistry of the compounds, the molecular properties of the compounds. And you can see here an example of different descriptors for different compounds. Here in the rows, you can see different compounds, in this case clustered by indication, clinical indication. And in the columns, you can see the actual descriptor, right? What this descriptor is doing is it's trying to capture the molecular properties of the molecule, right? And this is essentially what everybody is doing when we are trying to build machine learning models. We convert the chemical smiles into this kind of abstract representation or numbers. Now, we are in the era of big data. And for many molecules, we actually know much more than just the chemical structure, right? For many molecules, we have done assays already in the lab. These assays have been published in some articles. Some molecules are even in the clinics, etc. And therefore, there's a wealth of information that we can find in the public domain and in the databases that it would be nice to incorporate it also as features for our machine learning models, right? So the whole idea of the chemical checker is to try and capture not only the chemical information that we have for the compounds, but actually their target profile, the effect of the compounds in human cells, for instance, or even the clinical behavior of these compounds, right? So as you can see, now we have some molecular representations that is actually much more extensive, and therefore potentially it can help us in building better models with less data. We have a model in the Ercilia Model Hub that actually allows you to calculate these full chemical checker descriptors for any molecule of interest. And the question is, well, this is very abstract, but is it useful? Well, we can do many things with these large vectors. We can do projections, right? We've seen that. We can take a large descriptor and project it into 2D by using, for instance, PCAs or UMAPs. Those are classical techniques for 2D visualization. And then we can see whether those UMAPs make sense at all. So this is what we did here for roughly 10,000 molecules. Each dot in this plot is a molecule, and near, like, proximal dots mean that they have similar chemical checker descriptors, right? And I think it's very interesting because what you can see quite clearly is that in these 2D maps, molecules are very clearly organized by disease indication, right? So if I come with a new molecule and this molecule falls in this region here, a good educated guess will be that this molecule can be useful for oncology. If a molecule falls in this island here, a good educated guess would be that this molecule might be useful for infectious diseases, etc., 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 right? So those representations are actually a very good starting point for trying to predict what might be the biological or clinical uses of our compounds. Interestingly, the potential of these chemical checker descriptors does not stop here. Even if we look at different metabolites from different species, we can see also very good levels of organization. We can even look at the metabolites in different human fluids or organs. You can also see some kind of clustering, or we can even look at the fruit ingredients, and we can see that depending on the ingredient, we can see organization in different regions of the chemical space, right? So those are really, really like general purpose useful descriptors that we can use as good starting points for starting to make to build our machine learning models. I hope that was clear. This is by far the most abstract couple of slides. Now things are going to get a little bit easier again. We've shown already a little bit of like projections, but we do have ways of doing projections very easily using some already dedicated Ercilia Model Hub models. One example would be this one, where we are projecting directly into 2D space any molecule you might have, projecting it in reference of a commercial library, in this case called ChemDiff. So it's very useful. This is actually very useful because you can just take your compounds, project them using this model, and you will obtain as a result 2 dimensions that you can plot very easily. And this is very similar to the example that Jason already showed on the first day. where you can see separation, clear separation between in red natural product compounds and in blue synthetic compounds, especially using the PCA and the TS-NIP plots, right? So these projections, we can do them in basically one click using the Ercilia Model Hub, and are very, very useful to understand what is the diversity, what are the clusters that exist in our compound collections. Very good. Let's now move into the last category of models that we have in the Ercilia Model Hub, which is called sampling. Remember, in the case of sampling, we are trying to get new molecules or find molecules that are related to our molecule of interest. Here again, the most straightforward instance of this would be similarity search. And similarity search is something that many of you might have done already, trying to find similar compounds to your compound of interest in the literature. Now, the problem is becoming technically difficult because our chemistry databases are growing exponentially, right? When I started doing drug discovery a few years ago already, the databases that we could find were up to like a few million compounds maximum. And today we have databases in the public domain that we can search against. For instance, the NMIN real database that can be up to trillions of compounds, right? So the size of the databases that are available out there is growing at a very fast speed, and therefore we need methods to search these databases efficiently, right? So in Ercilia, we have some models, for instance, this one, that allow us to search against these ultra-large chemistry databases and do it very efficiently. Why is this useful? Well, because many times you have a good molecule starting point and you want to find some analogs that you want to directly purchase, for instance. So it makes sense to look for these similar compounds in chemical vendors so that you can purchase those compounds to do some tests. Interestingly, it is the pharmaceutical companies who have the largest databases, orders of magnitude larger than what we can find in the public domain. But unfortunately, those databases are not accessible to most of us, right? Pharmaceutical companies have their databases basically locked behind IP constraints. But even if so, we can search databases that are really, really large, like trillion scale today in 2025. So that's a very good way, before doing any generative chemistry or anything, to actually find analogs for your compounds of interest. But then, of course, we are in the age of generative AI, and therefore we also want to find methods for generating new chemistry, completely new chemistry that is not in any database whatsoever yet, right? And this falls within the realm of generative chemistry. And we do have some models in the Ercilia Model Hub, and we are still growing and incorporating more because this is a field that is evolving really, really, really fast. And I'm putting here an image that you might have seen or not. I don't know if you guys like this image or not. I find it awful, but it's very interesting, right? Because this image is a portrait of someone that was generated by an AI in 2018. And that portrait was sold in an auction for, I think it was $800,000. And why was that important? Well, because that was the first artistic painting that an AI was able to generate. And if you can see also the signature of the portrait is actually the name of the algorithm that was able to generate this portrait. So that was 2018, only 7 years ago. And if you see the images that we are able to generate with AI, for instance, using ChatGPT, the quality is incredible, right, compared to what we had in 2018. And the same things that we are experiencing with generative chemistry, right? We are able to generate very good compounds, and those methods are evolving every day. And every year we find methods that are able to generate better and better and better molecules, right? So what we have today probably is going to feel old or even archaic in a couple of years, right? But we do have some models in the Ersilia Model Hub, and I think that Gemma already nicely introduced this concept of reinforcement learning. Reinforcement learning is, I would say, the most commonly used method for generating compounds. The idea is that you have a tool that generates some hypotheses, so some compounds, and then there's an agent that evaluates whether those compounds are nice or not, and if they are nice, it rewards the algorithms, right? So you keep generating better and better and better molecules. That's the whole idea. We do have several models in the Ersilia Model Hub that are able to do reinforcement learning. I'm just showing one here, EOS 694W, where we input a few molecules and we ask the algorithm to generate completely new molecules that are related to my input, right? And it's quite interesting. I think it's quite plausible, right? Those molecules are not completely different, but also not completely similar to our input molecule. And you can tune this. You can tune this to generate like more diverse or more focused sets of compounds given an input molecule, right? So this is only going to improve. And this is definitely a tool that we should all be incorporating in the future, generative chemistry, if we are doing any medicinal chemistry or drug design overall. And yeah, sure. I just wanted to like reinforce the fact that generative chemistry is basically just the other side of the coin of what we have been learning, which is like converting compounds, predicting activity, right? Those are the annotations. When we talk about generative chemistry, what we are really trying to do is the opposite exercise. We have an activity of interest and we want to predict or generate compounds that are able to fulfill that activity, right? So generative chemistry and compound annotation are intimately related. Here you can see an example of a generative round that we ran for this initiative called Open Source Malaria. You can see that we asked the algorithm to maintain a core and then make some derivatives here in the branches, right? So as the reinforcement learning algorithm was improving, it was generating better and better compounds with higher predicted activity, right? So this is how those methods work. You always couple the prediction of some property of interest to the generative process. And we did that from a laptop computer, actually, and we were able to generate hundreds of thousands of compounds. So that's quite remarkable. Here you can see a map, all of those compounds. You can zoom in this map and find each dot is one of those compounds, one of those analogs that we can then cherry pick for further investigation. Here in red are the ones that have highest predicted activity, and those were, of course, like the ones that we ended up selecting. We teamed up with some medicinal chemists. We suggest, I believe it was 8 compounds, out of which 4 were active below the micromolar. This was an antiplasmodial assay, and the baseline or the expected activity of this chemical series in particular was below 1%. So the fact that out of 8, we obtained 4, that is 50%, that had a plausible activity, was actually quite remarkable, right? We increased the hit rate of our assay quite remarkably. And this is exactly what we want to do with this kind of tools, right? We want to generate compounds that are enriched for the activity that we are looking for. Yeah, it's never going to be perfect. Not all compounds will be active, but at least the ratio of compounds that are active with respect to the ones that we test is much higher, right? This is really the goal of virtual screening and generative chemistry in drug discovery. All right, so don't think that generative chemistry is easy. This is not something that can be done in just one click. It needs expertise, it needs tinkering, it needs playing with it, it needs basically just being skeptical about what you get and being critical about what you get, right? And we've been thinking a lot about this problem of like why navigating the chemical space, that is finding the good molecules in this huge, vast chemical space that we are dealing with. Why is it so difficult? And I think that the reason or the challenge that we are facing is exactly the same challenge that the Romans were facing. This is a map from the Roman Empire, where you can see kind of like the profile of the Mediterranean Sea, and they called it the Mare Nostrum back then, and this is an approximation of Europe. And I have to say that they did a pretty good job, right? And they did a pretty good job. Why? Because they sampled Europe a lot, right? They had many instances, they had many experiments, they were navigating this sea, and therefore they had a very good idea of how the landscape looked like, right? However, when you look at other parts of the world, for instance, Africa, they got it completely wrong, right? And why? Because they didn't explore it that much. And this is exactly the same problem we are facing when we are doing generative chemistry. It is very easy or fairly easy to do generative chemistry for known chemical classes for which we already know a lot about the activity or the QSAR, right? But it is very difficult to do generative chemistry for completely new chemical classes, especially for diseases that are underexplored. And unfortunately, diseases that are underexplored are many times the ones that are of interest to us. For example, neglected tropical diseases, right? Or infectious diseases for which there's not a lot of experimental data yet, right? So we need to be aware of these challenges. Generative chemistry is not always going to work equally well because we need some data in the background to help us in this navigation of the chemical space. Yeah, I hope that was clear. Very good. All right, so I'm going to pause for like a few seconds. Are there any questions? Anything in the chat? Gemma, Jason, all good? All right, perfect. Now, does AI for drug discovery stop with small molecules? Definitely not, right? When we think about drug discovery, we really need to think about whole systems. Yeah, it's not just about the small molecule, it's about the small molecule in the context of a biological system, right? And this is a beautiful painting by David Goodsell depicting E. coli, right? And you can see that the complexity is enormous, right? We have proteins, we have lipids, we have genetic material, we have compartments, et cetera, et cetera, et cetera. And small molecules are just a very tiny part of the whole system we need to be taking into account if we really want to discover fine and effective drugs, right? So, of course, AI is all over the place today, and there are methods and there are tools that are trying to also help us in addressing this complexity from scratch. And I do think that it's important that we also talk a little bit about it, even if we might not become experts in it, right? Because our expertise is more like in designing small molecules, etc. I think it's important to be aware of the complexity and how people are trying to model this complexity using computational tools. And it's quite remarkable. You can see here how people are using computational tools able to assemble some models of whole organisms, right, that contain, as I said, the membrane, contain the genetic material, contain the proteins, etc. And those models, I think that this is what's important, are built by analyzing the individual parts in isolation. And this is the important thing here, right? Because our experiments are and will always be snapshots of different layers of biological complexity. And our goal is to assemble those observations into something that makes sense. Yeah. And I like to think about this problem as basically making a fruit salad where you have the different types of fruits. Each fruit could be a different type of experiment. For instance, this would be a proteomics experiment. This can be a genomics experiment. This can be a metabolomics experiment. This could be a microscopy experiment, high throughput screening, whatever that is, etc. And then, of course, what we have to do is to come up with ways of, with the computer, building this fruit salad in the proportions that are right and in a way that makes sense, right? So this is what really people are trying to do in the age of AI. We, of course, also try to do this to some degree. We believe that the best way of tackling this huge complexity is by leveraging this concept of biological networks or graphs. Here is an instance of a biological knowledge graph that we built a few years ago, where you can see that we have the different types of, let's say, biological entities. And at the center of biology, we have genes, obviously, genes and proteins. And you can see that genes interact with many other types of entities. For instance, genes are expressed in different cells. Cells assemble to form tissues. Tissues might be related to diseases. Genes are also related to diseases. Genes might have functions, participate in pathways, have functional domains, etc. And genes, importantly, interact with compounds, right? Compounds can treat diseases, compounds can cause diseases, etc. So this is a complex network that we really want to use as a framework to help us understand this biological complexity we have to deal with in drug discovery. And I guess that what's important here is that each edge here in this biological knowledge graph corresponds basically to one experiment, right? An experiment where we measure whether a compound interacts or binds to a protein would be an instance of this edge in particular, right? So it would be one edge here in this network. An experiment where we measure the genetic association between a gene and a disease would be an instance in this edge, etc., etc., etc. So now we have a framework to basically just screen all the databases, all the scientific literature that we have out there, and try to populate a huge network that we can use to start tackling the complexity that we have to deal with in drug discovery. Yeah, and this is just an instance of this network where we had, I believe it was like 50,000 experiments collected. Now today, this network is I believe is like something like 300 million edges, right? So this is growing and growing and growing, and this is really our starting point when we try to do predictive biology and try to predict what is the effect of compounds in living systems. Okay, so we focused a little bit on compounds, basically throughout the course. Now I just want to give you a little bit of an overview of what can we do from the perspective of AI about genes or proteins, yeah? And when we talk about proteins, when we talk about genes, but especially proteins, I think AI has a lot to say because proteins can be, as you will see now, understood as language, right? So let me pause for a second and talk about this AI tool that we have all been exposed to in the last few years, which is called ChatGPT, and just explain very briefly, in very lay terms, how ChatGPT is trained. ChatGPT is trained based on a corpus of text. This text is extracted from the web, and basically what the authors of ChatGPT did is a very simple thing. They take this text and they mask parts of this text, right? And then they ask ChatGPT to complete this. So this says a time ago in a far away, and is asking ChatGPT to complete this by a long time ago in a galaxy far, far away, right? So when you do this over and over and over and over for millions, trillions of sentences, of documents, etc., you end up having a model that looks like it's intelligent, right? Because it's able to complete text, right? And this is exactly what you do when you interact with ChatGPT. You give some text and you ask it to give you more text in return, right? That's how those models are trained. So they seem to understand language because they've seen a lot of language, yeah? Exactly the same can be applied to proteins. Proteins can be described as a sequence of what? Of amino acids, right? We have 20 natural amino acids, and we have many sequences out there. We have hundreds of millions of sequences that people have been collecting over the years in databases, right? So we can take exactly the same algorithm that we have been using for ChatGPT and mask amino acids from the sequences and ask our model to complete this sentence, that is this sequence, right, of amino acids. And the more sequences we show, the more the model is going to learn essentially the evolutionary, the functional, and the structural patterns that are captured within this sequence. I hope that makes sense, right? ChatGPT was learning the semantics, the lexicon, the meaning of sentences by seeing many sentences. This is learning the evolution, the structure, and eventually the function of proteins just by seeing many proteins and understanding how they are related to each other, right? Okay. And then here is where basically the magic happens, because as you start doing this, you have sequences and you learn how to reconstruct masked sequences. You get an algorithm that is actually very intelligent and understands a lot about the underlying principles of protein biology in a way that it's actually able to predict how the proteins fold. And this is great because understanding how to go from sequence to structure and eventually to function is perhaps, or has been perhaps, the greatest challenge of molecular biology, right? And this is why last year, 2024, the Nobel Prize was awarded to the inventors of AlphaFold, who were actually able to predict protein structures with accuracy given protein sequences, right? And today people can use these algorithms, be it AlphaFold or any other, to actually visualize the protein universe in a way that was not possible before, right? Basically just trying to fold all of those sequences and give us or yield a map of protein structures that we can browse and navigate, right? And this is incredible if we are doing drug discovery, because having protein structures is really what we need to start designing compounds that are very specific to our targets of interest, right? So for the field, the good news is that for the field of global health in general, that's transformative, because many of our organisms of interest, for instance, pathogens, were neglected or understudied. And now all of a sudden, we have plenty of predicted structures for our organism of interest. You can see here just an example, right? And some quantification. For instance, for schisto, we now have like 13,000 protein structures, and we didn't have many before, right? So of course, those are just predictions. Of course, those structures might have problems. They might need to be like fine-tuned, et cetera, but those are really, really good starting points if we are doing structure-based drug discovery, right? So this is how AI is really penetrating into our field, by making predictions of a lot of the biological building blocks that we have in biology. Now, things are becoming even better, especially during the last year, because those those protein folding, we call them protein folding methods, have now been expanded not only to deal with amino acids, but actually to consider all atoms, right? So why is that important? Because when you start considering biological entities in an all atom resolution, you can start merging them, right? If we talk about all atoms, it doesn't really make a difference if we are dealing with proteins, or we deal with RNA, or we deal with metabolites, etc. Right? So we now have models that we call them multimodal, that can allow us to deal with proteins, small molecules, nucleic acids, etc., at the same time, right? And this is, of course, very important for drug discovery, because at the end of the day, what we want to do in drug discovery is to see how a protein structure interacts in complex with a small molecule, right? And now this can be done with one single model, right? And there's many instances. One is called Rosetta Fold. AlphaFold 3 is also able to do that. One that is called Boltz2 is also able to do that, etc., etc., etc. And we will start seeing more and more and more of those in the next months and years. I hope that is clear. Yeah. So structural biology is now at a point where AI-driven structural biology is now at the point where it's hopefully going to be able to predict not only protein structures, but also protein structures in complex with ligands. Very good. So I don't know if you guys have seen The Hitchhiker's Guide to the Galaxy. It's a great movie and great book, especially. And I think that the motto of The Hitchhiker's Guide to the Galaxy is, yeah, we are entering a new galaxy, but please don't panic. Everything's going to be fine. Nobody's going to lose their job, right? Many of us have been experimental scientists. Many of us wonder whether AI is basically just going to surpass all of the experiments, and everything will be able to be done in an automated manner with these AI tools. That's definitely not going to happen. Expertise will be needed more than ever in the age of AI, because it is very important that we are able to guide those systems and to critically assess what those systems are doing. And therefore, the expertise that we all have here, it's going to be very, very crucial. Yes, so we will all be using AI in the future. Definitely, yes. Nobody's going to lose their job because of AI. Definitely not in medicinal chemistry, not in drug discovery, not in biology research. Even if so, there's a lot of things that are going on in terms of trying to automate our processes in the lab, right? And here we just wanted to show a couple of examples of really interesting tools that are kind of like agentic systems, as we call them, that are able to do many things in the lab. For instance, this is something called ChemCrow. ChemCrow is just an AI, a language-based AI, that is able to do different actions that are necessary for the normal functioning of a synthetic chemistry lab. For instance, ChemCrow is able to propose a synthesis plan, is able to find for the reactants in commercial libraries, is able to tell you whether some of those reactants might be toxic, and is basically then able to execute a robot that is going to perform the reaction for you, right? And then it can do some literature search, it can do some annotation of the compounds, etc., right? So those kind of systems are great assistants for us, right? They will make our life easier in the lab, but they are not here to replace us. Definitely not. Yeah. ChemCrow is a good example, although it's like very early stage. Just a couple of months ago, there was a paper in Nature, also very interesting, where this lab in Stanford, they basically showed that they can extend this kind of idea to generate new SARS-CoV-2 nanobodies, right? And that's very interesting, because the way they did that is they created different AI personas. For instance, an immunologist, for instance, a computational biologist, for instance, a scientific evaluator, for instance, a principal investigator, for instance, a postdoc, etc. And they've put all of these AI personas to actually interact critically amongst them, so as to come up with a good scientific plan that could then be executed, right? And they successfully did that to some degree, and they were able to generate some nanobodies that were active against SARS-CoV-2, right? So also this is a direction where we are going. Does this mean that people in this lab were not working? Actually not, on the contrary, but they were interacting with these so-called assistants, right? So I think that this is more or less the direction that we will be experiencing in the next few years. All right. So in summary, I have exposed a little bit or extended upon like what has been exposed in the previous 3 days about this Ercilia Model Hub repository. The Ercilia Model Hub, you've just seen a tiny bit of it, but contains many, many models, software tools that you should definitely explore if you want to do any computational drug discovery. As a reminder, those tools can be useful to annotate your compounds, that is to know the properties of your compounds or the potential bioactivities of your compounds. Those tools can be used for molecular representation, that is to build or to calculate descriptors of your compounds if you are an advanced user, or to visualize the chemical space. And finally, those tools can also be used to sample new compounds, be either via like chemical similarity or with generative chemistry tools. The Auxilia Model Hub is something that we are just making now, right? So any feedback, any request, anything you might find more or less interesting, any bugs you find, anything, you should definitely let us know, because this is not a tool that has been published yet. We are building it and we want to make it like the best possible. So your feedback will be super welcome. Okay. Ercilia is focused on, as I said, like compound annotation, molecular representation, and chemical space sampling. But if you have more ideas, please reach out to us. This is only the beginning. We believe there's a lot of things that can be incorporated in the Ercilia Model Hub, not only for small molecules, but as we have seen a little bit for proteins and genes, which do have a lot of potential for AI-based modeling. Very good. Okay, I'm going to pause here. I don't know, Gemma, Jason, Nicola, if there are specific questions in the chat. How am I doing time-wise? What's the time? All right.
You're doing great. Okay. You're doing
Yeah.
great, Miguel. And I don't think there are any major questions we should answer in the chat right now. All questions are being collected and we're answering them in the background, and we'll share them after the session.
Okay, fantastic. Thanks, Nicola. So now, I mean, we are perfectly aware that there hasn't been like a lot of time for Q&A in the previous days, and the reason is because we wanted to allocate or group or cluster the questions that you all have been asking to today's session, right? So now I'm gonna open it a little bit for all of us to ask questions, right? Anything really. There's no good or bad questions. Directly related to today's session or to previous sessions, it's now the moment to ask those questions. Please use the chat, and we'll try to address them. Having said that, we have clustered a little bit in topics based on what you have been asking during these days, and we'll try to keep a little bit of some structure, right? So try to ask your questions related to the topic that I'm currently talking about. And we have about 15-20 minutes to talk about this. So please use this opportunity to ask your burning questions. So the first topic, or the first kind of questions that we have really been seeing in the chat these days, and we would like to address them now, is about the applications and the limitations of AI in drug discovery. And some examples would be something like, what are the current and future applications of AI in drug discovery? And what are the key limitations? For instance, data scarcity, bias, lack of generalization, and how do we address them, right? So I'm going to try to summarize this in an answer that more or less covers all of this. But if you want more specific clarifications, please feel free to ask. The applications of AI in drug discovery, I think, broadly speaking, for our interests, fall in three categories. One is trying to use models that are available out there to predict activities of compounds of interest. Yes, we have seen a lot of those in models that are available in the Ercilia Model Hub, but we have definitely not seen a lot of those in other types of predictive tools that are more structure-based. Yeah, those would be like, let's say, docking-like exercises, right? So this is definitely something where AI can help in trying to predict whether a compound is able to inhibit a pathogen or be toxic or have some desired or undesired properties, as well as predict whether a compound is going to bind to a specific target or not. That's a clear application of AI, yeah? Where you don't have to do much because those models are already out there. The second application of AI is when you want to train your own models, yeah? is when you want to train your own models. In that case, basically what you need to have is a labeled data set, right? And you should reach out to an expert. We haven't learned here how to train models really, right? And this is not something that you can do very easily. So you should find someone, perhaps in your institute, perhaps a collaborator that is knowledgeable about how to train AI models and see whether they are able to model your data sets of interest, all right? And then finally, the last application of AI is definitely going to be in the chemical sampling, right? Trying to predict new compounds based on a property or a initial compound of interest. This is very important, obviously, for hit to lead optimization stages, but also for the novel drug design. Okay. I'm going to pause here. Gemma, are there any questions? Any?
I can ask one on behalf of the chat.
Oh, sorry. I think Gemma was speaking, but not in mic. Right, Gemma? Okay. Jason?
Just because this is very relevant to our audience, do you have any models that can be used for the natural product chemical space?
Yeah, that's very relevant. Thanks for the question. We do have, I think we saw yesterday on Gemma's session, I think we saw a model that is able to kind of estimate whether a compound is more natural product-like or not. That's one thing that you should definitely check if you're dealing with natural product compounds to see whether your compound actually looks like a natural product or not, right? Because some natural products are more synthetic-like and some are more like natural product-like, right? Definitely, we do have those compounds. But the question is not really this, right? The question is whether you can apply any of the models that are available out there to your natural products. And this, unfortunately, I have to answer that this really depends on the model. If you have time, always look at the publication related to the model and see whether that model was built on a large chemical collection or a small chemical collection, right? If it was built on a large chemical collection, it is likely that this model is going to be useful for your chemical space without much problem. For instance, the cytotoxicity models, I would definitely try them with natural product compounds, right? And see whether the results that you get are plausible. So models built like on perhaps smaller data sets that might not be so applicable to natural product compounds, but it's always worth trying. So what I would do in this case is if you have some natural product compounds and you already know some activities of them, you can check, for instance, from the literature, you can check whether the models that we have are able to predict those activities to some degree. And if they are, perhaps then it's worth trying to run those models against natural product compounds that are not annotated yet. Okay. Yeah. Gemma, is there another question? No, we can move on if you want. All right. So the key limitations, this is something that, of course, people always ask about. And there are many, right? In our opinion, the main key limitation is data scarcity, and especially related to this idea of applicability domain, which is highly related to the previous question. Applicability domain means we have trained a model on a particular data set. Is it going to be useful for my data set of interest, right? So in general terms, the larger the data set for training, the more applicable my model is going to be to new molecules in general, right? So data scarcity is and will always be a problem or a limitation of machine learning, and we all need to be aware of this, right? If a model doesn't work for your molecules, doesn't mean that the model is bad. It might just be that it's not applicable to your chemical space. Models have some biases, right? A clear bias of models is when you train them on imbalanced data, right? we train them on many negative compounds, which is typically what we find in drug discovery, many negative and just a few positives, then those models might become a little bit biased toward just predicting negative compounds, and they might miss some positive, right? We do our best to correct for that, but sometimes this happens. Generally speaking, basically just lack of generalization, which is very related to this idea of applicability domain. So how do we address these problems? Well, part of it is something that you don't have to really worry about. This is the task of the model builder, to make the models as good as possible. But you, as users of the model, should always be critical, right? You should always critically assess your predictions. You don't have to test to trust them blindly. You should try to test whether the predictions are actually useful to you. So the best way to do that, if you have the capacity, is to do some experiments. You choose just a few compounds, right? You make the predictions, and you then go to the lab and see whether those predictions actually make sense. Yeah. So that's the best way of addressing those problems, actually going to the lab and doing some experiments.
There is a question maybe interesting here, which is what is the infrastructure needed to establish an AI ML lab?
Very good. What is the infrastructure needed to establish a machine learning unit, right? So look, in our experience and in our opinion, infrastructure should not be a key limiting factor. There are many solutions today to get started with AI. Most of the large language model providers provide free tiers for using those AI tools, so that should not be a problem. And when it comes to the Ersilia Model Hub, the infrastructure that you need is actually just conventional computers, no GPUs either, right? And we even provide like some cloud solution, as you have seen, to run some small predictions online. If you just want to predict a few compounds, you can use the online tool. If not, the best way is actually to download those models and deploy them locally. But for local deployment, you really don't need like any specific high performance computing or GPUs. Most of the models, thankfully, today in drug discovery can actually be run in conventional computers. Any more questions? All right, I'm going to move to the next kind of cluster of questions, which was related to especially like the challenges in data collection and in model validation, right? Very related to what we have been talking about already, right? So as you will notice, if you start doing data science and machine learning for drug discovery, as you will notice, data are typically very sparse. Many times it's incomplete, and many times it's unstructured, right? Also, many times it's very scarce, especially for natural products or neglected diseases, right? So dealing with this complexity is something that we just need to be comfortable with, right? How do we handle this? Well, here again, the best way, if you really want to do serious data science in drug discovery, the best way is to find computational chemists, computational biologists next to you that can actually help you, right? You need to speak more or less the same language. Hopefully now you are able to do it after this course a little bit, but it will require some programming skills. It will require some statistics skills to actually understand whether your data is complete noise or is actually informative, right? So answer to this question, don't do it alone. Try to team up or collaborate with people that are actually experts in data engineering and data processing. So what defines clean data, right? Well, defining clean data it's actually not possible. Although everybody wants to know whether their data is clean enough or whether their data is sufficient, right? In general terms, our recommendation is that you try to clean the data up to a point where all molecules that you have in your data set are valid. They can be processed with any conventional chemoinformatics tool. That's the first thing. And the second thing is that your outcome variable, right, your label, for instance, your activity is binarized in active, inactive. Yeah, if you reach to this point, then this is really a good starting point for machine learning, right? So clean, valid molecules, binary outcome. That's really what any machine learning expert would like to have to start building models. Okay, and how do we validate the models afterwards, right? So when you have a model, how do you validate it, right? We've talked about this already a little bit. The best way is to do some experiments. Other ways of doing that is to taking some held out data in a cross-validation setting, as Gemma showed already, and just you build models on a small part of the data, and then you make predictions on the held out data set. And if the model seems to perform well in the held out data set, you can cross your fingers, and the hypothesis is that it's also going to work in new data prospectively. Okay? Very good. So another group of questions that we have been getting are related to, like, how do we design models? What are the best features to use? And what are the best algorithms to use, right? So broadly speaking, I would say that this is the task of the machine learning expert, right? And until you guys become experts, I think that this is something that you should do in collaboration always. But you can participate a lot, right? And in particular, if you are a medicinal chemist or you have like deep knowledge of like the biological assays, I think you can be really, really helpful in designing the best possible models. For instance, the intuition of the medicinal chemist in selecting the best molecular descriptors, right? Whether stereochemistry is going to be critical or not for the activity, whether solubility or other physical chemical properties are going to be crucial or not for penetration through the membrane. These kind of things, it's really what the machine learning modeler needs, because that's going to determine what are the descriptors that they will use to build the models, right? Then it was also asked, like, when do we use supervised, when do we use unsupervised, and where do we use reinforcement learning? This was answered already via email, I believe, but I think that generally speaking, supervised learning, you start doing this when you have training data. So when you already have some results from your experiments, you can train a model that then you can use for new molecules. Unsupervised learning, this is typically used when you just want to find clusters in your data set, find how your data set is distributed in the chemical space, basically evaluate the diversity, right, of your compounds, but you don't necessarily have any label data or anything, right? You just have a collection of compounds with no bioactivity annotated whatsoever. And reinforcement learning, this is not something you want to do from scratch. This is difficult to do. It's related to generative chemistry most of the times, and therefore you should do reinforcement learning in collaboration with a machine learning expert, right? This is why we have not given like hundreds of examples on reinforcement learning in this course, because this is just an intro, right? And then also, how do we evaluate critically, or how do we understand all of those concepts of like model performance that we find in the papers, right? Well, the first thing that you really want to check is for measures of accuracy, and we find like different evaluation metrics. We've talked about precision, true positive rate, area under the ROC curve, sensitivity, recall, all of that, right? So all of those metrics, you should study them and understand why are they useful and why we need to take into account more than one, right? There's no one single answer. Depending on the scenario, we might want to do something or the other. So understanding all of those concepts is going to be very useful for you to really go through the machine learning and AI papers that are being published. How to select thresholds, this largely depends on your needs. If you can only afford doing a few experiments, right, you want models that have high precision. If you can afford to do more experiments, maybe you want models that have high recall, right? This is more or less the rule of thumb. And how to avoid overfitting? Well, this is the job of the machine learning expert. The best way of avoiding overfitting is training with more data. But if this is not possible, you need to play with the model architecture. And this is definitely not something that you can do yet, right? You need to continue like learning a little bit of machine learning principles to start like being able to tinker with the training procedure and the choice of the model algorithms. Any questions in the chat? No?
I think we are good.
Okay. All right. And then there was another cluster of questions that we believe is related to interpretability and explainability of the models. That's very important and is a fair concern, right, that many of us have and many reviewers have when we submit machine learning models, which is like, okay, these models are very complex, but this comes at a price. The price is that those models many times are perceived as black boxes, right? And this is not good. It's not good because if we cannot interpret those models, if we cannot know what's happening inside those models, it's going to be very difficult for us to trust those models first and then to improve them, right? Or to use them consciously. So there's a whole field of research related to explainable AI or interpretable AI that is definitely very interesting and is definitely improving for drug discovery. Broadly speaking, what people are trying to do is to find algorithms that are able to highlight either one, the physicochemical properties of the compounds that make them more active or less active according to the model, or two, highlight substructures, functional groups in the compounds that are responsible, quote unquote, for the predicted activity, right? And this is, of course, both of things are very important because this is very actionable knowledge for the medicinal chemist, right? If you know that, for instance, solubility plays an important role, and this is something that the model is telling you, then you can start doing some SAR playing with the solubility, right? If you know that the particular chemical moiety is responsible for the activity, then you might want to preserve that chemical moiety as you improve your compounds, et cetera, right? So explainable AI, definitely yes, people are aware of this limitation of machine learning models in general, and people are trying to develop better and better explainable AI tools. All right, and then in general, basically, people were interested in basically trying to understand how to get started, how to use more tools, how to use more platforms, and basically knowing the landscape of tools that are available out there today, right? So generally speaking, we have tools that are readily available for the academic sector, and those are or should be open source tools, or at least free to use tools. There's many of them out there. The Ercilia Model Hub contains a compendium of them, but there's more. And you should definitely look into what tools people are using in the papers. And if you find them interesting, try to use them, right? And then there's another family of tools, which are like more private sector tools that are licensed and you have to pay for them. Having said that, many times you can get free licenses if you are an academic researcher, right? So yes, of course, there are many tools. The Ercilia Model Hub is very focused on antibiotic drug discovery. For other fields of research, there's other tools that might be also interesting to you, right? And how do you get started? How do you get started in AI if you are a beginning? And what are the most valuable skills to contribute in AI ML-driven drug discovery? In our opinion, the best way of getting started is becoming a user, not a model builder. It is only when you start using tools that are out there that you will become familiar with what are the strengths and limitations of those tools, and eventually become someone who will be able to develop their own models. Yeah, so start being a user, start trying those tools. They are not perfect. They will produce bad results sometimes, but sometimes they will produce good results. So start understanding them, and then you will be able to start developing your own tools. If you end up developing your own tools, you will have to know So some programming, right? Specifically Python, and this takes some time. So just be aware of this, right? Python is more or less the standard language for data science and AI/ML today. So any developer that is doing AI/ML in drug discovery and anywhere else for that matter needs to know some Python. That's one path. The other path is if you guys have a good data set that you have been producing in your lab, a data set that you have the intuition that can be valuable for training a machine learning model. In that case, you should reach out to someone who is able to build a model, because probably your data set is going to be very valuable and people will be willing to collaborate with you. Okay? So it's either becoming users or either collaborating with someone because you do have a data set that might be worthy for machine learning model training. Okay? Very good. I think I'm running out of time. I'm going to leave it here and hand it to Nicola.
Okay, thank you so much. So you've reached the end of the course so far. And I think there's been fantastic engagement, a lot of questions. And now really what it's about is about drawing everything that you've learned and starting to integrate it into your life. So I'm going to take you through a brief reflection session now, where I'm going to ask you to write. The first step is going to be to write some ideas down. So let me show you what it looks like. So I'm going to give you maybe a little bit longer minutes of writing, and you're going to write a list of 3 to 7 or more, if you like, things that you learned from the course. All right, so these are things that you've changed somehow in the course of this week between Monday and Thursday. What are 3 to 7 things that you've learned from this course? And you're going to write them either on your laptop or on a piece of paper, or you don't have to put them in the chat just yet. So this is just for you. Okay, so your minute starts now. I'm going to time you. Off you go. So you should be writing 3 to 7 things that you've learned from the course. You're halfway there. Last 10 seconds. All right, so of course you might have learned some more, but just for the purposes of getting your brain going and into the zone, that's all the time that you had. All right, so now you're going to choose. Look at that list and think, what are the top 1 or 2 things that I really got from this course, that I really achieved? So what are the top 1 or 2 ideas, concepts, tools, skills, awarenesses, thoughts, whatever it might be? And I want you to put a little star next to them. And now you're going to share them with the group if you're up to it. Okay, so now you're going to write them in the chat, the top 1 to 2 things that you learned. You're going to write them in the chat, and let's see what comes through. Jason, are you up to reading a few out?
Sure. I'm waiting for the first to come through. Synthesizability prediction. It's the first one. We'll give some people a few more seconds to start typing. How to select the best models for my work. Yeah, that's a very key one. Supervised and unsupervised machine learning. Criteria for selecting the best drug, applying AI models to real data sets, the basics of how AI is developed, data cleaning, more supervised and unsupervised learning, reinforcement learning, so the different types of AI, and generative AI. I see a few. Whew, they're coming fast now. Thick and fast.
I think we're going to enjoy reading these after the session. These look really, really fantastic. It's great to see some concrete ideas coming through. I'm just going to give another few more seconds to see some more coming in. And feel free to like or send little hearts to any ones that you particularly like of other people's. Although I know they're coming super fast. Beautiful. Beautiful, guys. Okay, so as you have typed and you're welcome to continue typing, we're going to move on to the next step. And now you've learned these things, and now how do you actually integrate them into your life? And so you can use this tool, perhaps you know it already, of SMART goals. So now I want you to think, okay, I've learned this thing, right? What am I going to do with it? Now I go back to my normal life. How am I actually going to integrate this into my life in some way? So I want you to come up with maybe 3 to 5 is a little bit much for right now. It might be more like 1 to 3 SMART goals that you're actually going to apply in the course of the next 6 months. But I don't want you to use only 6 months. So your first goal might be a shorter time period, maybe a week or a month. Your second goal might be something like 1 or 3 months. And your third goal might be something like 6 months. Okay, so that's the time, the amount of time that I want you to give kind of to each goal. You're going to be really specific about what you want to achieve. So when you've done the action, you want to know, I've done it. Okay, I've done it or I haven't done it. It's got to be concrete. It's got to be measurable, right? Something that you can, you know that it's clear that you've done it, right? There's a target that marks your success. So for example, if it's a simple one like you're going to read a publication, once you're at the end of that publication, that's measurable, right? You've read every word. Okay, so that's an example of something that's measurable. It's got to be achievable. It can't be something crazy like tomorrow I want to start an AI lab. Okay, no. That's what, a 15-year, 10-year, 5-year goal? Okay, I'm not sure. But it's got to be something that is possible to accomplish within the constraints that you currently have. It's got to be relevant, so it's got to be connected to your work, and it's got to be connected to this webinar series. And as mentioned, it's got to be time-based. Okay, so what might these look like? So some examples, right? You might decide to clean a set of data and run it through the Ersilia Model Hub within 2 weeks. You could even do that this afternoon, right? And then you've done it, but you're giving yourself 2 weeks. That's achievable, okay? It's time-based, it's all the things, right? It's all the things. And that might be a goal that you choose. You might want a slightly different way of looking at the measurability so that it's not words that you read or data sets that you clean, but it might be time. So spend 30 minutes a week exploring the Ersilia Model Hub and running new evaluations. So this is rather you're going to carve out 30 minutes on a Thursday afternoon or something, put it in your calendar for the next 4 weeks. And in that time, no distractions, nice little snack, you're going to start to explore the Ersilia Model Hub. Okay, and that's how you're going to start to build this knowledge. You might decide to complete the Introduction to Small Molecule Drug Discovery in the next 3 months. That's manageable. It's a 17-hour course. Or you might decide to read one relevant publication per week for 4 weeks and discuss it with a colleague. So, and I see a lovely WhatsApp group has started amongst the participants. Maybe you're going to join that and start a little reading group or something like that. Okay, so I'm hoping by now, while I'm talking, you've got maybe one goal that you can share with the group. So we're almost out of time. So now I'd like you to write one of your goals in the chat. So remember, you've got to have a time specification. It's got to be measurable. You'll know if it's relevant to your work. You'll know if it's something that you can achieve. So let's see what you've come up with. What? And don't worry if it's humble, and don't worry if it's grand. Okay, these goals are for you. So it's up to you to choose what they are. All right, Jason, Gemma, Miguel, can you read out a couple from the chat as they come through?
So we have learn the basics of using machine learning models for predictions, finding out more about machine learning models. We have a short goal, educate others about the Acilia platform. Long-term goal, collaborate with others who have access to drug molecule datasets to come up with a relevant outcome.
Nice. So some of these are smaller and some of these are massive. So that gives you a nice lot of scope.
Yeah, have a read of publications that created models that align with his work within 1 week. Make some predictions for Plasmodium falciparum using the Acilia platform in the next 2 months, I think it is. Go through the recordings and learn more about using the Acilia Model Hub. So refreshing concepts. Doing the drug discovery course. Read 1 publication a week and share with friends.
I love that. Your friends.
Yeah, you will become scientists too.
Yes.
Select a data set and clean it and start a new project of finding a lead compound with optimized properties over the next 3 months.
Fantastic. Okay, let's leave it there. Keep filling the chat. Feel free to keep filling the chat. We'd love to hear and to be able to read your goals. But I'm going to move on to our list of upcoming opportunities that you might like to be aware of. So here are some opportunities and just things to have in your mind, to have in the ether. Okay. So of course we've really punted the Introduction to Small Molecule Drug Discovery and Development course via Coursera. If you choose the option without a certificate, then it's free. Okay. If you want to get the certificate, then I think there's a fee. But yeah, it's a great course, and even without the certificate, to get you into the world of drug discovery, it's going to be very useful. Then there's the H3D Foundation webinar series. I've shared all the links to that, but I'll share them all again. We load everything onto our YouTube channel, and also there are about another 12 webinars coming up this year. So please join the mailing list, and then you'll be updated, and they're on our LinkedIn and everything. And then just to be aware, there's the H3D Foundation Symposium on the 13th to 16th of October next year. This is very much by application. We've got a limited number of bursaries and things like that. There are no calls for applications yet, but just keep a lookout on our socials and see what comes up over the course of the next few months. And then if you don't know already about the Grand Challenges African Drug Discovery Accelerator, it's a closed group. There are about 32 members at the moment in Africa, but I want you to know about it because it's going to be relevant to your work as you go forward. It's a group about developing the capacity in drug discovery and development. It's a network, and it's supported very much by the logo below it called Rafiki, which is an EU-African alliance, and we run training programs, mentorship programs, and all sorts of interesting things for the members of GCADA, Grand Challenges African Drug Discovery Accelerator. So these are not open calls, unfortunately, at this point, but I want them on your radar because as you move up in this career, these things are going to be interesting to you. Okay, so just be aware of that. All right, and then to connect with us, we've got lots of ways. Acilia has a mailing list, and their LinkedIn platform is incredibly active. Of course, their website is fantastic as well, so you can explore all the links there. And H3D Foundation as well. There's a mailing list, and our LinkedIn platform is equally active. So just be aware of those places to connect with us. All the information, all the opportunities are shared on those platforms. So do connect with us there. Last but not least, we'd like to thank our funders, the Schmidt Sciences, for making this course possible and for making this project possible. I'd like to thank my colleagues who've just brought their whole hearts to this project and to this training program. And finally, to thank you on behalf of all of us, to thank you for joining us, the participants. It's been amazing to have you with us, to have your engagement, and to walk beside you on this journey. And we're very excited for all the interest that's been generated by our offerings. So I know some of you have emailed us during the week. give us a minute. It's been a busy week, but Miguel, Gemma from Acilia will connect with you whenever they can, and from HDD Foundation as well will be as connecting as possible within the amongst of all the things that we need to do. So thank you again. I'm going to stop sharing now so we can log off. I don't know if my colleagues would like to say any final farewells as we end off the session.
Yeah, I just want to say thank you to everybody. It's been a lot of people here, and you have been basically all coming every day, which is remarkable. It's a lot of content, it's a lot of new stuff. But yeah, I mean, it's great to see so much engagement, especially via the chat. And hopefully you guys got the first quick overview of what AI can do for you in the future. And yeah, let's just stay in touch, of course. Thank you.
Maybe just to add, yes, it was a lot of content, so I would recommend go through the notes. Everything will be made accessible afterwards. Rewatch the recordings. It's a lot of concepts to learn in just a few hours. So yeah, please don't feel overwhelmed. Just go through it step by step, and as you start applying it to projects, it'll make a lot more sense. So yeah, great engagement as well. So thanks.
Hi, everyone. Yes, so thanks for your attendance. It was great to be able to deliver this course and to see the interest in the tools that we are developing. I cannot stress enough the importance of going back and revising the concepts to really get started with a solid foundation in AI. We've got some direct emails, and we will answer all of you as soon as we can. Ercilia develops these tools and does research and partners with scientists everywhere in the world, particularly in Africa. So we aim to answer to all of you within the next coming weeks, but please have patience. You don't have to email me 3 times. I will respond as soon as I can, okay? So thanks for your patience, and looking forward to staying connected.
Thanks, guys. And to end off, just to say, yes, there is one more task, and that's the feedback form. And this is important, really important for us, but it's also important for you. So it is your final task of the course. So please complete the feedback form. I will email it to you, but I've put the link in the chat as well. And of course, it's been uploaded onto your SharePoint of all things AI2050, which is on Acilia's Git book. So yeah, make sure you complete that. And certificates will be out by the end of the month. So just give me a bit of time to complete those and to consolidate all the information. And the end of the month, you should be receiving a certificate. Okay. Thank you so much, everybody. Great session. We loved having you, and see you next time.